All right, well, welcome back. We're on the home stretch now. We're on the last of the material for the course. So we're starting the material for the third test today. And of course, then this material will be on the final as well. So I'm recording this during spring break, so I don't actually know how the results of the second test went or were, but hopefully it went well. Um, so anyway, moving on to so from here we're starting with blood, then we do the rest of the cardiovascular system, and then we get into the lungs, the digestive system, and then our last section is on metabolism. So as far as blood goes, the first stuff is pretty basic and you're probably fairly familiar with it. So the primary function of blood is to transport, and specifically to transport oxygen. So that's what the O2 is on this slide. And then, in addition to transporting oxygen, of course, blood is also going to transport waste products. So you could consider carbon dioxide one of those waste products, as well as nitrogenous wastes, which are the byproduct of breaking down proteins. So remember, we've talked about uremia, which is the buildup of urea, and how that can be toxic to the central nervous system. So we have to be able to get rid of those nitrogenous waste products, and so that's one of the things that the blood does, is helps us get rid of those. Of course, in addition to oxygen, the blood transports other things that we need. So obviously it transports fuel like glucose or like free fatty acids. It also transports proteins to get those to the cells that are going to use them to repair themselves or to grow, whatever it is that those cells need to do. And then from chapter 16, the most recent chapter, we also talked about how blood is used to transport hormones. So things like insulin, glucagon, parathyroid hormone, and many others. So those get dumped into the blood, and then the blood transports them throughout the body, and then whichever tissues are target tissues, because they have the receptor for that particular hormone, then they'll cause an effect in those specific tissues. But in addition to transport of different substances, the blood is also responsible for regulation of both temperature and pH. So one of the things that we're able to do is to absorb and distribute heat with the blood. So we'll talk here in a case study in a little bit about one of the ways that we manage our core temperature is we direct blood away from the core, so away from your abdominal organs in particular, and we open up blood vessels near the skin surface. We give up some of the fluid component of the blood and also some additional extracellular fluid. We give that up as sweat, and so then we try to cool ourselves off through evaporative cooling. So being able to absorb and distribute that heat is an important job for blood. Another thing it does is it maintains the acid-base balance of the body, so it maintains the pH. So one of the things that blood does is to act as a reservoir of bicarbonate ions. So bicarbonate is basic, and so it's where this comes up is when you exercise intensely. Obviously, you're going to produce lactate, and so then that drops the pH of the muscle, makes the muscle more acidic. And so, in order for the muscle to be able to continue to contract, we export that acid into the bloodstream, and then it can be used in other tissues. So, for example, the heart is really good at breaking down lactate because it has lots and lots of mitochondria. And so, one of the things that the blood does is it acts as a buffering system. It helps us keep our pH within normal limits in the body. And in addition to that, blood serves that protection function. So it has antibodies and white blood cells in it that help protect us against invading pathogens, whether those be bacteria, viruses, or even worms. And we'll talk about the specific cells that protect us against those. So in terms of the basic characteristics of blood, so as you know, the color of blood changes a little bit depending upon whether or not the blood is oxygenated. So essentially, blood kind of runs the spectrum from this really bright red color in fully oxygenated blood to a sort of darker red, kind of a maroon color in deoxygenated blood. And you might have noticed that if you've ever donated blood or if you've had medical testing done where they took a vial of blood, you probably noticed that it was really maroonish kind of a color. It's a fairly dark color. So that is deoxygenated blood that comes from the veins. So remember that there are two sets of blood vessels. There's arteries and veins. Arteries take blood away from the heart. Veins bring blood back to the heart. And in the vast majority of cases, arteries are going to carry oxygenated blood and veins are going to carry deoxygenated blood. So when you go and give blood, they're going to take that blood from a vein. So you're giving up deoxygenated blood. So you probably noticed then that it has kind of that really dark red color. So a question that comes up sometimes, although I haven't gotten it at Whitewater, to everybody here's credit, I did get it at a previous institution. I had a student in a similar class like this one raise her hand and she said, okay, well, so if blood's red, how come my veins are blue? And so I didn't think she was serious, but she was. And so the reason for that, in case you were curious, is that the veins appear blue because the overlying skin, fat, and fascial layers. So obviously you've got layers of tissue atop the veins. So they are fairly superficial, but there are tissues that sit on top of them. And so those other tissues scatter most of the red portion of the white light before it can reflect off of the blood. The blue light, or the blue part of that spectrum, is reflected, and that's what bounces back to our eyes. So the fact that all of those overlying tissues, the skin, the fascia, and the fat, scatter that red portion of the spectrum, and then blue is what gets reflected back, that's what gives why those veins appear blue when you look at them, is actually because of those overlying tissues and the scattering of the red component of the light spectrum. And it's a really similar idea to what we see at the sunset, where the sun is going to appear red because of the scattering of the blue part of that light spectrum by the atmosphere. So the blood is red, not blue, but the shade of red varies with oxygen status. You can also see there that it's alkaline, which means that it's basic. So its pH is slightly above 7. So again, it plays a role in helping us buffer acids, so particularly lactate that's produced from muscle action. It's also viscous, and viscosity deals with thickness. So in terms of it being viscous, it's thicker than water. And the reason that it's more viscous than water is because it's got lots and lots of formed elements in it. So it's got lots of cells, like red blood cells, white blood cells, platelets. You've got some glucose dissolved in there, amino acids, fatty acids, etc. And so with all of those things dissolved in the blood, that makes it thicker, right? So something that is more viscous is a thicker fluid. And actually, for what it's worth, the viscosity of the blood changes depending upon hydration status. So if you are dehydrated, then the plasma component of the blood, which is the fluid component of the blood, decreases. So the blood becomes relatively more concentrated. And so that more concentrated blood, where you've got the same number of red blood cells in less solution, that more concentrated blood is more viscous. It's a thicker fluid, and it becomes more difficult for the heart to pump it. As opposed to, if you're really well hydrated, then you're going to get an increase in plasma volume. So you're going to get, again, same number of red blood cells, but with a lot more fluid that they're floating around in. So the blood is effectively thinner or less viscous. So a more viscous fluid is thicker, a less viscous fluid is thinner. In terms of blood volume, that varies depending upon body size. So in general, men have somewhere between 5 and 6 liters of blood. Women have between 4 and 5 liters. But the reason for those generalities is that men are generally larger. And so because men tend to be bigger, men tend to have a higher blood volume. So there are other things that affect that other than size. So training status and heat acclimatization are two of the most important factors that affect that. So if you are endurance trained, so if you take up a running program, what you're going to see is that you actually get an increase in blood volume. So initially, you're going to get an increase in plasma volume. So you're actually going to retain more fluid. And what that does is it decreases the workload on the heart a little bit. You get a little bit thinner blood, and so it makes it easier for the heart to beat that blood or to pump that blood around the system. Now, eventually, you're going to produce a few more red blood cells. That production is going to catch up so that your hematocrit, which is a term we'll talk about in a second, but so that the hematocrit stays about the same. The other thing that can change blood volume, so endurance training can increase your blood volume. Something else that can change your blood volume is heat acclimatization. So it's a very similar response to what we see with endurance training. If you're used to being out in the heat, if you work outside all the time in Florida in the summer, 90 degrees probably doesn't feel hot to you. And so part of the reason for that adaptation is, some of it's in the hypothalamus, but from the blood standpoint, you're going to hang on to extra plasma volume so that you can give up more fluid to sweat so you can keep yourself cooler. So people who are better conditioned tend to have that higher plasma volume, they tend to be a little bit more resistant to heat. But not only that, when they're exposed to either exercise or heat, they start sweating more and they start sweating earlier. But their sweat is more dilute, so it has less sodium in it, for example. So that's actually, again, part of those heat adaptations and part of those endurance training adaptations is we try to keep our core temperature as close to 98.6 as possible. And so if you're used to being hot or you're used to exercising, you're going to start sweating fairly early with the onset of exercise, and you're going to sweat quite a bit. Now that said, some people are just heavy sweaters, so just because you sweat a lot doesn't mean that you're necessarily in good shape. When I was in grad school, one of my colleagues was a pretty big guy who played high school football, but he hadn't probably worked out in at least 6 years. And so walking around in the summer in Texas, he just sweated profusely. And we took a physiology class, and in the class, the professor talked about what I just mentioned, that if you're in shape, you sweat more and you sweat earlier. And so he turned around to the rest of our cohort and was like, "Look, I'm in shape because I sweat all the time." So you can just be somebody who sweats a lot. And then there's also, for what it's worth, some people are salty sweaters, and other people are not. And so how much salt that you give up is also dependent upon, to some extent, genetics. So that may be one of the features of your own cooling system that you might have noticed. All right. So in terms of the major components of blood, so the two primary components are plasma and formed elements. And again, plasma is the fluid that everything else floats in. So plasma is kind of a yellowish or straw-colored fluid that is about 90% water. So there's some other stuff in there. So there's obviously oxygen and carbon dioxide dissolved in there. Now, that's not the primary way that we transport oxygen, but it is the primary way that we transport carbon dioxide. You've also got glucose, fat, proteins, sodium, other things dissolved in there, but plasma is 90% water. So the way to think about it then is that's the fluid that everything else floats around in. And the everything else are the formed elements. So the formed elements are the cells, for the most part, not platelets, but everything else there is a cell that floats in that plasma. So your erythrocytes are your red blood cells. So that's what RBC stands for. So those are your red blood cells. And the hematocrit, the term that you see below erythrocytes, hematocrit deals with what percentage of your blood is comprised of red blood cells. So in men, the average is 47%. So what that means is that of your total blood volume, 47% is red blood cells. But that varies. It's kind of plus or minus 5%. So if average for men is 47%, well then the high end of the normal range is 52%. That would be pretty exceptional and fairly unusual. But the high end of the normal range is 52%. And then the low end of the normal range is more like 42%. So for women, their hematocrit is a little bit lower. For women, their normal hematocrit averages around 42%. And then with plus or minus 5, so it can get up to 47% or down to 37%. So one of the things to notice there is that the primary reason for that difference actually doesn't have to do with stature, as many of the other things we talk about will. The primary reason for that difference is actually related to testosterone. One of the things that testosterone does is increases red blood cell production. So men tend to have a little bit higher oxygen carrying capacity because we have, in general, more red blood cells. So remember that red blood cells, their primary job is to carry oxygen. So the more red blood cells you've got, the more oxygen you can carry. So the more oxygen you can deliver to the tissues, so the more energy you can make aerobically. And so that then, the more red blood cells you've got to a point offers a performance advantage. So again, hematocrit is the percentage of your blood that is red blood cells. So what you're seeing in the picture is spinning down blood, centrifuging it, and separating out the plasma, which is on top there, the yellow portion, as opposed to the red blood cells, which drop to the bottom. And then the buffy coat, those are your white blood cells. So you've got white blood cells that also are in your blood, and we'll talk more about those at the very end. And then you've got platelets, which assist in clotting. All right, so as far as the structure of the red blood cells go, so you'll notice, so that's you've obviously got top view and a side view there on the right side of the slide. They're kind of flat discs. They sort of look like a donut with the hole filled in the middle. So they don't have a hole in them, they're filled in, but it's sort of narrower there through the middle. So there are some important things about the structure of the red blood cells, because remember there's an important interrelationship between structure and function of everything that we're going to talk about. And so as far as red blood cells go, that shape is going to be really important. And in addition to that shape, they are fairly springy. They have a really resilient cytoskeleton, so they're capable of being squeezed down to a really small, or basically being compressed as they squeeze through really small capillaries, and then they can spring back into shape once they've gotten through those capillaries. So the fact that they've got a really resilient cytoskeleton is important. The fact that they're flat is actually really important, because remember, their primary job is to carry oxygen, and so oxygen is going to move with concentration and pressure gradients. So it's going to move, as we'll talk about, primarily from high pressure to low pressure, but it can't move very far. And so we want to minimize the distance that oxygen has to diffuse. So if we have a nice flat red blood cell, well, that minimizes the distance that the oxygen has to move from the inside of the cell to the outside of the cell. If the red blood cells were big circles, then that would, some of that oxygen that's kind of in the middle of the cell, that's that much farther it has to move, and it would have a problem. It would have a hard time diffusing out of the cell. So the fact that it's a nice flat disc enables the red blood cells to offload or onload at the lungs the oxygen more easily. It facilitates oxygen transport. You've also got no organelles in your red blood cells, so there's no nucleus, there's no mitochondria, no Golgi apparatus, none of the other stuff that we talked about. They're basically just tiny little bags of hemoglobin. And so hemoglobin is what actually transports the protein, and we'll talk about that more in a second. But the reasons that matters, first, the fact that they have no nucleus, the reason that's important, remember that your nucleus is your instructions for building proteins. And so because they have no nucleus, they can't make proteins, and thus they can't repair themselves. So your red blood cells have a fairly short lifespan. It's typically only 3, maybe 4 months. And the reason for that is because as they go through those tiny little capillaries and get compressed and then spring back, eventually those proteins are going to wear out. So the proteins of the cytoskeleton get worn out, and so they can't fix them. And as a result, ultimately the cell is going to lose its springiness. It's going to get torn up, gets kind of degraded, and then it gets usually it either gets picked up in the spleen, or white blood cells will eat them up outside of the spleen. They'll phagocytize them is a more scientifically way of saying that. So no nucleus means no repair, means a short lifespan. If they don't have mitochondria, well, then they can't use the oxygen that they're transporting to make energy. So how do they make energy? They do it anaerobically. So they break down only glucose, and they do it using the glycolytic enzymes. So they can break down glucose without the presence of oxygen. And so that's how they make energy. So the good thing about them not having mitochondria or a nucleus or anything else is any of those organelles would crowd out space for the hemoglobin, so they wouldn't be able to transport quite as much oxygen if they had organelles taking up space. But if they had mitochondria specifically, they would be using up some of the oxygen that they're transporting. So to avoid that, they don't have mitochondria, and they work strictly anaerobically. So those are the important structural characteristics. And then in terms of the functions of the red blood cells, so again, the primary function of a red blood cell is to transport oxygen. They do transport some carbon dioxide, but most of that is transported in the plasma, and we'll talk about that when we get to the lung chapter. And so as mentioned, they are pretty much just tiny little bags of hemoglobin. And so you can see a hemoglobin molecule pictured for you there. So there's 2 components to that. So there's globin, so there are 4 globin chains. So there are 2 on top, those are the 2 beta chains, and then 2 on bottom, which are the 2 alpha chains, and each chain has a heme group that's attached to it. So that is the iron-containing component of hemoglobin, is the little heme group. And so each heme group binds to 1 molecule of oxygen. So each molecule of hemoglobin then, with its 4 globin chains and thus 4 heme groups, is able to carry 4 oxygen molecules. So a single red blood cell has somewhere in the neighborhood of 250 million hemoglobin molecules, and because each hemoglobin molecule can carry 4 molecules of oxygen, each individual little red blood cell can scoop up about 1 billion molecules of oxygen. So each little red blood cell can transport quite a bit of oxygen, and then you've got, I believe it's trillions of red blood cells. It's a staggeringly high number. So we're able to transport quite a bit of oxygen when we need to be able to do that. All right, so erythropoiesis is the production of red blood cells, as you can see there. And so remember that takes place in red bone marrow, and red marrow is primarily in the axial skeleton, and specifically it's in the flat bones. So remember that in adults we have red marrow in the scapulas, the sternum, the ribs, the pelvis, and then the skull primarily. And so that's where we make most of our red blood cells. So every day you churn out somewhere in the neighborhood of an ounce of new blood, and then you make in that blood about 100 billion new cells every day. So we make lots and lots of red blood cells every single day because, again, they have a fairly short lifespan and they wear out. So if we need to make 100 billion new cells every day, we've got to have the component parts to do that. So the component parts are the normal macronutrients. So you've got your carbohydrates, your fat, and your protein. Obviously, you need sufficient amounts of those. But there are also a couple of additional, or actually 3 additional special nutrients that you need in order to make red blood cells. So the first one on there is iron, because again, in order to have the heme groups for hemoglobin, you need iron, and you need sufficient iron intake. So there's 2 types of iron in terms of dietary iron that you would consume. There's heme iron and non-heme iron. Heme iron is found in animals, and non-heme iron is found in plants. And the difference matters because in non-heme iron, you're not able to absorb very much of it. So specifically, in the small intestine, you're able to absorb about 2 to 5% of the plant-based or non-heme iron that the small intestine sees, so very little of it. And so you get that non-heme iron. Typical sources or good sources of it are oatmeal, spinach, beans, and lentils. But if you can only absorb 2 to 5% of it, that's not very much. You need to eat quite a bit of spinach to consume enough iron in order to keep up with the requirements for making 100 billion new red blood cells every day. So a better source in terms of being able to absorb the iron is heme iron, which comes from animals. So rather than absorbing 2 to 5% of that, in the case of heme iron, we absorb somewhere between 10 and 35% of that. So again, that comes primarily from animal products. So you get that from places like beef, liver, if you're into liver, pork, tuna, and clams. Also, if you're into clams, those are good sources of iron. So one of the things that comes up sometimes is that vegetarians may have anemia. They may not be able to produce enough red blood cells. And so part of that is related to insufficiency or insufficient intake of iron. And again, it may not be that they're not taking in enough total iron because they're taking in, eating lots of spinach, beans, etc., that have a decent amount of iron in them. It's just the fact that we're not really able to absorb that very effectively. That said, there are some ways to increase that. So particularly vitamin C is effective at helping us absorb more iron. So for example, the amount of vitamin C in 1 glass of orange juice can increase absorption of iron in the intestine by up to 3 times. So that can make a pretty significant difference in iron uptake. On the opposite side though, fiber, which obviously you're going to find in spinach and beans and most of the other places where you have non-heme iron, fiber inhibits absorption, as does calcium, because calcium and iron are going to use the same transporters in the intestine. And so they compete. So the other 2 things on there are vitamin B12 and then folic acid. So both of those are B vitamins, and they're both needed in order to carry out normal DNA synthesis. And so if you're making lots and lots of new cells, you have to replicate lots and lots of DNA. Now, I said a minute ago that the red blood cells don't have a nucleus, and that's true, they don't when they're mature, but when they're developing, they have a nucleus, and then they eject it right before they mature. So the immature red blood cells do have a nucleus, and so to make lots and lots of red blood cells, you need to reproduce DNA relatively rapidly and relatively consistently, and so you need vitamins B12 and folic acid in order to do that. And if you have a deficiency of either of those things, can't make enough DNA, and so any cell that turns over really quickly, you have a tough time making enough of. So you'd also see that in some of your epithelial cells if you have a real deficiency. Obviously you turn over like the lining of your stomach, the lining of the intestines, the lining of your larynx fairly frequently, and so you'd have some some problems there as well, but it's most pronounced in the red blood cells just because of the sheer volume of cells that we have to make every day. So in terms of places that you find those things, so B12 is found primarily in animal products, just like heme iron. So you find that in places like fish, beef, poultry, milk, eggs, and other milk products. And then folic acid is found primarily in vegetable sources. So in your leafy greens, your citrus fruits, beans, breads, rice, and pastas, because the pastas are oftentimes fortified, meaning that it's added to them. So another reason that vegetarians sometimes are anemic is because B12, again, found primarily in animal sources. So if you combine that fact with the fact that heme iron is absorbed more effectively, then they don't get the easily absorbed source of iron, and they don't oftentimes get enough B12, and so that combination of things can lead to an anemia or an inability to produce a sufficient amount of red blood cells. And then destruction of red blood cells, so I mentioned, typically happens, they last about 3 to 4 months, and again, it's related to their inability to repair themselves. All right, so speaking of blood, so I have a case study for you, or kind of a thing. So Lance Armstrong, as with many of my examples, it's getting a little bit more dated these days, quite possibly a lot more dated. But to give you some background information on Lance Armstrong, you're probably familiar with a couple of different things. One is that he was a good cyclist, and two, that he cheated. And so my question is, how did he win so many races? Well, to give you an idea of what he did, between 1999 and 2005, he won 7 consecutive Tours de France, right? So won that thing 7 consecutive times, the pinnacle of cycling. And the Tour de France itself covers 2,200 miles over 21 stages on 23 days. So you get 2 days off over the course of 23 total days. The other 21 days you're riding, and you're covering more than 2,000 miles. Just being able to do that alone is amazing, and being able to do it really fast is even more so. To give you a little bit more background on me, so many of you know, or most of you know, that I grew up in the Austin area. I was an undergraduate at the University of Texas between 1999 and 2003. So I was in college as an undergrad when Lance Armstrong was winning the first of his races. And there's a big cycling population in Austin, and so I was always amazed talking to people who were cycling enthusiasts and people that were on like the cycling club at UT, how often people genuinely believe that he just won because he worked really hard and that he had really good genetics. And both of those things are true. Certainly he worked really hard and he does have really good genetics, but cycling since its inception has been one of the most drug-laden sports out there. For example, so in the late 1800s, competitive cyclists were known to take heroin, cocaine, and ether-soaked sugar tablets. And so ether at the time was a surgical anesthetic. It wasn't a very good one, but it reduced pain a little bit. And so if you took a pain reducer with some sugar, then that could obviously help you cycle farther and faster. Not surprisingly, with that cocktail of heroin, cocaine, and ether, the first doping-related death in cycling occurred in 1886. So again, doping was pretty prevalent in the sport for more than 100 years before Lance Armstrong ever won a Tour de France. And it wasn't just cyclists. So there were runners. At that time, they were known as pedestrians. But runners of the late 1800s and early 20th century also used alcohol and strychnine to help their performance. So the idea behind alcohol is that it numbs pain. And so, for example, in one of the early Olympic marathons, I think it was the 1912 games, there was an athlete whose nutrition plan, if you will, was to take a shot every mile. And I believe it was a shot of whiskey. And he made it to around the 12th mile before he finally had to bow out of the race, which nonetheless is impressive. Like, I can't imagine doing a shot a mile for more than 10 miles and still being able to run in a semi-straight line. Strychnine, the other thing that I mentioned that they would use, you might have heard of that one. It's found now in rat poison. And so what it does in high doses is actually paralyzes the breathing muscles of the rats. So their intercostal muscles keeps them from engaging in their normal rhythm of contraction and relaxation. And so because of that, then the rat asphyxiates. If you take strychnine in low doses, though, it causes muscle fasciculations, causes twitching, and so the cyclists and runners who took it, you'd get that muscle twitching, and so they thought, oh, the muscles are excited, essentially, and so that's going to help me run or cycle faster, and so that's why people took it. Doping deaths related to cycling, so they're one of the most talked about, I guess. In the Summer Games in Rome in 1960, there was a Danish cyclist named Knud Jensen who died. There were some allegations that it was related to amphetamine use. It was probably at least in part due to heat, but certainly there was some drug use that was part of it. And then in 1967, a rider in the Tour de France from Britain named Tim Simpson died from amphetamine use. So all of that is to say that cycling has always been a sport rife with doping. So the fact that somebody could win 7 years in a row and not be doing that strains credulity. So the reason they won so many races, and again, it takes everything, right? It takes hard work, it takes genetics, but then it also takes something extra to be at that elite of a level in sports. So in terms of blood doping, the idea with blood doping is to get more red blood cells, because the more red blood cells you have, again, the more oxygen you can transport, and the more oxygen you can transport, the more ATP you can make in the cells aerobically. So you're not producing the byproduct of that, you're not producing lactate, and so you can just produce more energy for longer, and so it improves your endurance performance. So there's a few different ways of engaging in blood doping. One is transfusions. So you can transfuse somebody else's blood, but that's pretty unusual. Typically what athletes would do is to have their own blood taken out somewhere in the neighborhood of 3 months before the race, have that spun down, just like the picture I showed you a few slides ago, pull out the red blood cells, and then freeze those, and then you re-inject the plasma. And then, so over the course of those months, the body will sense that you have decreased oxygen carrying capacity because of the blood loss, and so you'll make new red blood cells to replace the ones that you lost. And so eventually you come back up to a normal hematocrit, a normal concentration of red blood cells, and then right before the race and during the race, you keep re-injecting those red blood cells, and so then that gets your hematocrit unnaturally high. So if your normal hematocrit is something like 48, then you can add red blood cells in there and you can get it, well up over 50. So that's one way it's done. Another way is erythropoietin. And erythropoietin is a hormone that controls the production of red blood cells, or more accurately, that stimulates the production of red blood cells. So there's certain cells in the kidneys that when they become hypoxic, when they sense that there's less oxygen, then they, through a signaling cascade, cause the release of EPO. EPO then stimulates the maturation and production of red blood cells in the red marrow, and so then you get more red blood cells. So you just take a hormone, again, it's produced by the kidneys, but you take a hormone and then you're going to produce more red blood cells. One of the reasons that people took that hormone, at least early on, is it was less of a pain when that hormone was developed. It was less of a pain to do that than it was to do the whole blood transfusion thing. You just take a hormone and you're done. But then a test was developed for that hormone, and so what do you do to get around the test? Well, you can just retransfuse your own blood. But that said, international cycling adopted hematocrit limits. So, and it's really hard to find what they are. My best guess is the hematocrit limit is somewhere around 52. So if your blood is higher than 52% red blood cells, then the assumption is that you did that artificially, that isn't just a natural gift that you have, that you've done that through blood doping or transfusions or EPO or something else that they haven't figured out what you've done. But the general idea is that there's no way you could have done that without cheating. So if you exceed the hematocrit limits, then you get kicked out. There's also something called synthetic oxygen carriers, and basically what that is a blood substitute. So at times cyclists use that as well, but as far as I'm aware, that's not quite as popular as the other two methods. And as mentioned, testosterone also stimulates red blood cell production. So Lance Armstrong, in addition to doing the various blood doping techniques, also took anabolic steroids. And so when people become aware of that, one of the questions that comes up is, looking at him, you're like, well, that doesn't look like a guy that takes steroids. When most of us think of somebody who takes steroids, we think of like a bodybuilder or a big football player, that kind of a thing. But because testosterone helps with muscle repair, so it helps stimulate the satellite cells which fix muscles, so it helps them proliferate, so it speeds up muscle repair. But in addition to that, it also causes you to produce more red blood cells. So testosterone alone is going to increase your ability to transport oxygen. Also, altitude helps with that. So if you go to altitude, so if you go to Denver or Mexico City, Denver is at about 5,200 feet, Mexico City is over 7,000 feet. And so when you go to altitude, your hemoglobin is less saturated. It's not as full of oxygen. So for example, and this has kind of come up with some of the coronavirus stuff, you might have used or seen a pulse oximeter. And so pulse oximeter, what it does, it's a little red light that they attach to the end of your finger, and it'll flash typically two numbers at you. The one number is your pulse, so somewhere in the 60s probably for most of the people in this class. And then the other number should be somewhere around 98. And what that is your percent of hemoglobin that's saturated with oxygen. So at sea level, and in a healthy person with no kind of lung disease, your hemoglobin saturation should be somewhere around 98%. Might be as high as 100, might be a little bit lower than that, might be like 96, but usually it's somewhere right around 98. With altitude, hemoglobin saturation rate is going to drop a little bit. So it's going to drop to, something like 96, 95, maybe as low as, probably not as low as 93, but somewhere in the mid 90s, let's say. And so the body's long-term response to that is, well, if all the red blood cells aren't going to be full of oxygen, what I'll do is just make more red blood cells. And so the effect then is that you've got more red blood cells and they're all partially full when you're at altitude, but if you take those red blood cells, take your chronic adaptation, and you then move out of the mountains, or if you go compete at sea level, now you have a bunch of additional red blood cells. So it's the same effect that you would have gotten from transfusion or EPO or testosterone, but you've done it by using an altitude chamber or by living at altitude. That said, if you train at altitude, and we'll talk about this more with the altitude chapter, but if you train at altitude, because of the reduced air pressure, your training intensity necessarily drops. You can't work as hard up in the mountains as you can at sea level. And so what you're supposed to do to maximize this effect is to live high and train low, is how it's usually phrased. So you live at altitude, so you're up there for 8 or more hours. Typically 8 to 12 is kind of what you need of reduced air pressure in order to see the adaptation. And then you train at sea level, so you live up in the mountains, then you drive out of the mountains for your training so that you can maintain your training intensity. But altitude has that effect of causing you to produce more red blood cells and helping you to transport more oxygen as well. So then the other question I have on there is what happens with too much? So one of the reasons that there is a hematocrit limit set by international cycling and other international sport governing bodies is because if you get too many red blood cells, well, then you increase the likelihood of clotting, because basically you got too many of them crowded in there together. They can bump into each other, they can get stuck to each other, and then they form a clot, and then they get stuck in some of your small blood vessels. So if they get stuck in the brain, it can cause a stroke, in the lung, pulmonary embolism, in the heart attack. So that would obviously be bad. It's also really difficult on the heart, really tough on the heart to pump blood that's that viscous or that thick. And so then it can also lead to, in that case, that would be heart failure. Basically from having high blood pressure, the heart having to pump too hard all the time. All right, and so this is that process that I showed you with EPO and what happens. So the thing all the way on the right there is the hypoxia, the low oxygenation of the blood. So that again would happen with something like if you went to altitude. That would be the decreased availability of oxygen, that third little bullet down. And so because of that, you got cells in the kidney that are sensitive to that are going to release EPO. EPO stimulates the production of red blood cells. And then once our oxygen carrying capacity is back to normal, then that removes that stimulus that is causing the kidney to release that EPO. So it's that negative feedback loop that we've talked about with hormones and in a variety of other places. All right, so I've got another case study for you. There's several case studies in this one. So the case study here is related to a high school football player from a school called Pleasure Ridge Park on the outskirts of Louisville, Kentucky. And in this particular scenario, Max is going through football practice early in the season. So it is 2 weeks into preseason camp, so it is probably mid-August, maybe starting to be late August or early September. And on this particular day, the practices occurred from approximately 4 in the afternoon to about 6. This particular day, it's 94 degrees. The coach of this team is a guy named Jason Stinson, and Stinson had been a former offensive lineman at the University of Louisville, so, in the same town. And basically, Stinson was pretty mad at his team. He felt like they were kind of screwing around in practice and not taking it very seriously. And so he got really upset, really frustrated with them, and he said, you know what? If we're not going to take this seriously, then what we're going to do is we're going to run until somebody quits. And so he divided up the team into two groups. You had the lineman group, so your offensive and defensive linemen and probably some of your bigger linebackers and fullbacks. And then you had more of the skill group, the small guy group. So you had, your receivers, your cornerbacks, your secondary players. And so then they ran gassers. And so what gassers are is you start on one sideline, run down to the other one and back, and then down and back again. So you run sideline to sideline twice. So it's a total of a little bit more than 200 yards worth of sprints in full pads. And so again, it's 94 degrees, running 200-yard sprints repeatedly. And so as mentioned, the coach Stinson told them we're going to run until somebody quits. And they ran reportedly for a little over 40 minutes. So that's lots and lots of sprinting. So after finishing the last gasser, so that last 200-yard sprint, Max, who is an offensive lineman, he was a larger guy, Max collapsed on the sideline. So shortly after he collapsed, he vomited and then was unconscious. His dad happened to be at practice, and so his dad and 3 assistant coaches ran over to him to try to assist him once he was unconscious. And in order to assist him, what they did was they basically took the buckets of ice water and dumped them over him. They took off his pads or his shoulder pads, and they took off his socks and then dumped the ice water over him. But in terms of what they didn't do, they didn't take him inside, they didn't put him in the shade, and there was no athletic trainer available. So a few minutes later, after doing that and he was not revived, they called 911. Max got transported to the hospital, and by the time he arrived at the hospital, his core temperature was 107. Keep in mind that brain damage can occur over 105.5 if you're there for more than 30 minutes. So at 107, you're probably starting to get some pretty significant damage. And so effectively what happened there, so Max was in the hospital for several days and ultimately passed as a result of complications related to heat stroke. But basically what happens there when somebody dies of something related to heat stroke, so remember that the blood, one of its jobs is to help us keep our core temperature down. So it's able to absorb heat and then it transfers it out to the periphery where we give up that fluid, and then through evaporative cooling we're able to keep our core temperature hopefully relatively low. But so essentially we've got 2 ways of cooling ourselves. One is through evaporative cooling, so that's through sweating, and the other one is through radiant heat exchange. And so you give off heat to the environment provided the environment isn't hotter than you are. And as an aside, and you're aware of this, the evaporative cooling is actually slowed down if you're in a more humid environment because the air can't absorb as much water off of you, and so you don't lose as much heat to that evaporative cooling. So between the actual outside temperature that day and the humidity, Max wasn't able to cool himself very well. And so ultimately what you end up getting there is the cause of death is typically listed as multi-system organ failure. And there's a sort of series of events that happens that leads up to that. So when you're engaged in intense exercise in the heat, what you're going to do is what's referred to as blood shunting. So you're going to direct blood away from the core. You're going to direct it away from your abdominal organs. So specifically or especially away from the kidneys, the liver, the small intestine, etc. And you're going to direct it toward the working muscles, and you're going to direct it toward the superficial blood vessels so you can give up some of that plasma volume as heat. But as you start to accumulate heat, so keep in mind, and related to something we talked about previously, so remember that a byproduct of ATP breakdown is heat production. And so the higher the intensity of the exercise, the harder you're working, the faster you're producing heat. And so even if you're well hydrated, if you're working hard enough and you're working in the heat and your ability to dissipate heat is impeded by pads and dark clothing, then you're going to accumulate heat faster than you can give it off. And so as your core temperature gets higher and higher, the heart becomes a less effective pump. You get what's referred to as cardiac hyperthermia. And so as the heart gets hotter and hotter, its output goes down, so you circulate the blood less effectively, which then you get less oxygen delivery to the tissues and less transport of blood to the skin to try to cool yourself off. Eventually, or actually coincidentally with that, so coinciding with that cardiac hyperthermia, you also get brain hyperthermia. So remember that the hypothalamus is responsible for detecting brain temperature, blood temperature, and regulating both blood pressure and that shunting, that direction of blood. And so as the heart and brain get hot, the brain gets less effective at sort of directing that blood shunting and keeping blood pressure up. If you exercise at that high temperature long enough, or if the core temperature stays high for long enough, then remember we've directed blood flow away from the intestines, away from the kidneys, the liver, etc. And so that ischemia, that lack of blood flow, can lead to the death of some of the tissue, some of that epithelial tissue in the small intestine. And so as that tissue dies, remember those epithelial cells in the small intestine, remember they've got tight junctions between them. And so the goal of those tight junctions is not to let bacteria and viruses and various things we've ingested pass into the bloodstream. Well, as some of those epithelial cells die in the small intestine, that allows bacteria to move from the intestinal tract into the bloodstream. And then it gets circulated throughout the body, and then you get a condition which is called sepsis or septic shock, which is essentially a systemic bacterial infection. And in addition to that, that exercise in the heat is going to cause the breakdown of muscles. They're going to release myoglobin, which we talked about, then can lead to kidney damage, and then you're also going to get renal failure that occurs there. So because the blood, one of its jobs is to help us distribute heat, it does that, and it does that typically fairly effectively, but again, that can be impeded by things like humidity and also the intensity of the exercise. So one of the things that I would want to convey to you here is that as it pertains to this stuff, remember that even if somebody, or I want you to know that even if somebody is well hydrated, they can still have heat stroke. Because again, if you're exercising intensely enough, if you're running through your ATP fast enough, you're essentially producing the heat faster than you can lose it through that evaporative cooling. And that's made even worse if the outside temperature, the ambient temperature, is hotter. So there's a related condition to exertional heat stroke, which is called heat exhaustion. And so basically that's where somebody is exercising in the heat and they just can't continue and they stop. And so that's oftentimes referred to as a safety break. Effectively what's happened there is that the hypothalamus has detected that temperature is hot enough, and so it causes you to feel excessively fatigued and you just stop. Another component of that is oftentimes, so I mentioned you can have any of these problems if you're normally or well hydrated, but oftentimes people have been exercising in heat for a while before they encounter these things. And so if you've also lost plasma volume, the blood is effectively thicker and it gets harder for the heart to pump it. And so then you just can't keep up with the intensity of the exercise and you have to stop. So it's usually this combination of cardiac hyperthermia and then the hypothalamus in the brain detecting that the core temperature is too high and causing you to feel excessively fatigued and causing you to slow down and stop. Those two are not a continuum. If you can get heat stroke, basically if you've gotten heat stroke, you've pushed right through exhaustion. So heat exhaustion is what usually stops you from getting to the heat stroke point because you actually stop exercising. What else was I going to tell you about that? There's something else. So one of the things with Max is that his parents sued the coach for reckless endangerment. That's a lie. That one was, that was reckless endangerment and gross misconduct. That's what they sued him for. And ultimately, the coach was not convicted. And then the reason that he wasn't convicted was the fact that Max was taking an ADD medication. I don't know if it was Adderall or Ritalin or what it was. But either way, those are amphetamines. And so one of the things that amphetamines do is they allow you to work harder without feeling like you're working that hard. It's referred to as reducing your perception of exertion. And so you can work harder, get your core temperature higher before you ever notice it. And we talked about this, different class. This class hasn't talked about that. Forget I said that. But with amphetamines, then you can work out harder and get your core temperature up higher before you ever notice. And so that certainly played a role in his death, that he was taking the amphetamines, because whereas most of us would stop at a core temperature of around 102, if you're taking amphetamines, you can get up to 103, 104 before you even start to notice that you're hot. So that certainly played a role in his death. And then the last thing on there is heat syncope. Syncope is fainting. And so effectively what happens when somebody has heat syncope, it's usually a really early season thing. If you're talking about like football or soccer, those fall sports. But effectively what happens is they're not used to being outside and not used to being in the heat. And so essentially all of their blood vessels open up near the skin, but their brain hasn't learned or figured out effectively how to close off the blood vessels to the intestines, to the liver, to the kidneys, etc. And so basically you have too many blood vessels open, which then reduces the amount of blood in the central circulation, which means the brain very temporarily gets a little bit less blood. And so with the brain getting less blood and less oxygen, you lose consciousness, and so they faint. And then typically when they're lying on the ground, they come back to within a few seconds. But that's heat syncope. It's because your body hasn't yet figured out how the whole blood shunting thing works in the heat. All right, one more case study for this chapter, and this is another football player. He's pictured there in the lower right. His name is Eric Gall. And so he was a sophomore football player at a school named Chadron State College, which is in northwestern Nebraska. And so he was there. He was a transfer player. He was originally from Florida, but he had transferred to Chadron State, and so it was his first day of football practice in August of 2016, and the temperature was in the high 80s the first day of football practice, so it was warm, but not excessively so. And so about halfway through practice, Eric started complaining of really excessive fatigue and lots of cramping, particularly in his legs, and eventually he collapsed. So he tried to fight through it as long as he could. Eventually he collapsed. He was taken over to the sideline, kind of carried over there. And so while he's on the sideline and the athletic training staff was talking to him, he complained of just being excessively tired, and that cramping continued, eventually working its way up kind of into his abdomen and chest. After a few minutes, he complained of really severe chest pain and then eventually difficulty breathing. So the athletic trainers called an ambulance at 11:16. It arrived at 11:21, and unfortunately, Eric was pronounced dead at the nearby community hospital at 12:30. So the question I have for you is what could have happened here? What would have caused this otherwise healthy football player to complain of really severe cramping, chest pain, excessive fatigue, and then eventually difficulty breathing? Well, an important thing to know, so you might be thinking heat, and again, it was in the high 80s, so it's hot, but it's not excessively so, and he's from Florida, and so he was fairly used to it. But another thing to know about Chadron State is that it's at 3,300 feet, so it's not high. It's not as high as Denver, but it's certainly not at sea level, so it is at a medium altitude. And so, in fact, that played a role in his death. And so what Eric actually died of was a condition called sickle cell anemia, which I would imagine you've heard of. So in the case of sickle cell anemia, what you've got is an abnormal hemoglobin. So this is the picture of hemoglobin that you've already seen. And so remember that there are 4 globin chains, 2 alphas and 2 betas. And so under really low oxygen conditions, as happens during exercise, especially exercise when it's fairly warm. So one of the things about hemoglobin is that it has less affinity for oxygen in warmer environments, so it's able to offload its oxygen more easily, which is good because obviously you get hotter when you exercise, so it's nice that hemoglobin is more easily able to deliver oxygen to the tissues. But if somebody has sickle cell anemia, what ends up happening is as you offload that oxygen, then the beta chains, so the top 2 chains on there, after they've offloaded the oxygen, they change shape and they form this really stiff and spiky shape. And then when that happens through all of the hemoglobin molecules in the red blood cell, then that changes the shape of the entire red blood cell. And so rather than being the normal kind of donut-shaped red blood cell you see there on the left, you get that sickle-shaped cell you see there on the right. And so, as mentioned earlier, as far as the structure of red blood cells goes, one of the important things about them is that they're able to kind of be springy. They can squeeze through small blood vessels and then pop back into shape. Well, if you've got that shape that you see there on the right, that sickled shape, that shape is not as resilient, and that shaped red blood cell is going to actually get caught in small blood vessels, and so you're going to get clots. You're going to get backups of blood flow in those blood vessels. And so one of the reasons that Eric was probably complaining of difficulty breathing is because he likely had a clot in his lungs. He likely had a pulmonary embolism. And so sickle cell attacks, the reason that they can result in death is you get those blood clots in the lungs, you get them in the heart and the brain and various other places, and so that's when they can become fatal. One of the interesting things about sickle cell anemia, so you can see there in beneath the pictures that hemoglobin, its amino acid sequence, its protein sequence is 146 proteins long, and that sickle cell trait is produced by a change in just one of those amino acids. So you can see there at the sixth protein in the chain, rather than being a glutamine, it's a valine, and so that one amino acid chain completely changes the individual's ability to adapt to high intensity exercise or deal with high intensity exercise, particularly at altitude. The reason that altitude matters is if you're at those medium and higher altitudes, again, the hemoglobin isn't going to be fully saturated, so not all those beta chains are full, and then high intensity exercise in the heat, you get significant offloading of the oxygen from the hemoglobin, and then the combination of those things between high intensity exercise, heat, and the altitude produces lots of offloading, produces lots of sickling, and so that cocktail of things is what tends to produce the attacks that we associate with sickle cell anemia. So it's a trait that's typically found in people of African descent and is found in as many as 8% of African Americans. Between 2000 and 2016, 11 NCAA Division I football players died as a result of sickle cell complications. So it's something that can be quite deadly and is certainly a high risk for certain athletes. So what it is, the reason that it exists, it's an adaptation to malaria. Malaria is a plasmodium that replicates itself inside of red blood cells, and so it's a useful adaptation in some ways, in that the red blood cells that would be infected with the malaria would assume that sickle shape, and so it kept the malaria from replicating. It basically buys time for the immune system to be able to attack that plasmodium and to recognize it. So it's a useful adaptation in terms of surviving malaria, but it's not the most useful in terms of that same sickling can be brought on by those other stimuli I mentioned a minute ago. So even professional athletes are affected by it. So Ty Montgomery, when he was playing for the Packers in 2016, had to miss time after playing the Bears because he found blood in his urine. And so he'd had effectively a minor issue with sickle cell anemia during the game, so he was ruled out for the following week after that. And then in 2007, Ryan Clark, who is on, I think, the NFL Network now. He does commentary for ESPN or the NFL Network or one of those. But in 2007, he was a safety for the Pittsburgh Steelers. And so during a game in Denver, he experienced really severe left side pain, and so they ended up rushing him to the hospital, and he had a blood clot in his spleen, and so he had to have his spleen and gallbladder removed. But he was able to return from that and to continue to play for 7 more years, but every time after that the team that he was playing for returned to Denver, he was deactivated, and so they replaced him so that they wouldn't risk a repeat of that incident. All right, so other blood disorders include anemias. So broadly speaking, anemia is the blood, the oxygen carrying capacity of the blood is too low. So there's a few different reasons for that. So the first one there obviously is blood loss. So if you get significant blood losses, you don't have as many red blood cells, you can't transport quite as much oxygen. So obviously blood losses inhibit your ability to carry oxygen, pretty straightforward. But another thing that can happen is you don't produce enough red blood cells, and there are a few reasons for that. One of those is iron deficiency. So again, as mentioned earlier, you might see that in vegetarians. You also might see that in just people that don't have a really balanced diet. I know adults that essentially live on French fries and macaroni and cheese, and so they're going to be probably iron deficient. So it can be, again, related to vegetarianism, or it can be related to just other dietary deficiencies. And that one's, iron deficiency anemia is usually pretty easy to correct. You just add some more iron into the diet. Potentially you add supplements, and usually that one solves itself. Pernicious anemia is a little bit different. So pernicious anemia is an autoimmune condition, and effectively your own immune cells attack the lining of the stomach, and so when they do that, they obviously damage it. They hurt that epithelium, and so they inhibit the stomach from producing some of the things that it needs to be able to produce, specifically intrinsic factor. So intrinsic factor is produced by the stomach, and you need that. So once things leave the stomach, they move into the small intestine, and we absorb most things in the small intestine. And so in order to absorb B12, which again you need for DNA replication, which you need for red blood cell production, in order to absorb that B12, you need the intrinsic factor that's produced by the stomach. Well, if you have an autoimmune condition where you wiped out those cells in the stomach that produce intrinsic factor, now you can't absorb the B12, and because you can't absorb the B12, you can't make red blood cells. So this condition was discovered or really elucidated in the early 1900s, and the discovery of it is fascinating. So effectively, they knew of iron deficiency anemia in the early 1900s, and that if you gave people food that had, if you fed people liver, if you fed them beef, those kinds of things that were rich in iron, that would oftentimes cure anemias. But they couldn't figure out with people who had pernicious anemia, they didn't really know what was causing it. They tried to give them high iron foods, and it still wouldn't fix the problem. And so a physician named George Minot in 1926 actually won the Nobel Prize for figuring it out. But in order to do that, he ran an experiment with people that had pernicious anemia, and he tried giving them various things, so various dietary interventions to try to get them to produce more red blood cells. So the things that he fed them range from kind of gross to horrific. So the first group, they fed chicken liver. Obviously, that's high in iron, but iron isn't the problem here, so the chicken liver didn't alleviate the anemia. Another group, they fed half-cooked ground beef, figuring that maybe it was the cooking process that caused the problem. Of course, that didn't solve it. They gave another group raw hog stomach, which helped a little bit, because then you'll get some of the intrinsic factor, but not much. And so what really eventually solved the problem was that they gave people who had pernicious anemia the regurgitated stomach juice of one of their grad students. So when you're thinking about going to grad school, that's the kind of thing that grad students may be called upon to do. So one of the grad students had to regurgitate some stomach acid. And then, I guess to be fair to Dr. Minot, they didn't just feed the patients straight stomach acid. They also, they spiced it up a little bit. They added butter, lemon, and parsley to it to make it a little bit more palatable. But at any rate, with that stomach acid, which would contain the intrinsic factor, they were able to alleviate the pernicious anemia, which again won Minot the Nobel Prize. So give people some stomach acid and solve the problem. Another one on there is too many red blood cells destroyed. So you can get that with autoimmune diseases, but for the most part, so it's essentially autoimmune or genetic diseases. That's usually where you see that there are too many red blood cells destroyed. But more interesting to talk about, more fun to talk about from a sports standpoint is polycythemia. So polycythemia is an abnormal excess, too many of red blood cells. And so what that does is it increases your blood viscosity, increases the thickness of blood, but it also increases the blood's oxygen carrying capacity. So the individual pictured there, the skier there in the lower right, is a Finnish skier named Eero Mäntyranta. Probably grossly mispronounced that. But he competed in the Olympics between 1960 and 1972. During that time, he won 3 gold medals, 2 silver medals, and 2 bronze. But one of the things, probably one of the primary things that helped him win all of those medals at the Olympics in those 12 years is the fact that he had an inherited genetic abnormality. And the particular condition that he had is called primary family congenital polycythemia, or PFCP. And what that does is causes you to make way more red blood cells than normal. So as mentioned, average male hematocrit is about 47%. So somewhere around 47% of the blood is red blood cells. For Eero, his hematocrit would be somewhere around 70. So he was basically blood doped all the time. And so because he had so many more red blood cells, he had just an enormously high aerobic capacity and enormously high ability to deliver oxygen to the tissues. And so he was a really exceptional endurance athlete, particularly in skiing. So he's brought up oftentimes when people talk about the level playing field and the idea that we're all equal in sports. Some people point to athletes like Eero and say, well, we're not all equal because you can have this sort of genetic alteration that effectively makes you a world-class athlete just based on that one gene mutation. All right, and then the last thing is our leukocytes. So those are our white blood cells. So the order that they're listed for you there is the order in terms of most to least of them in the blood. So as you know, your white blood cells have a really important immune function. And so there's one of the important differences between them and the red blood cells. The red blood cells just carry oxygen, but the red blood cells also stay in the circulation. They dump oxygen out of the red blood cell, and it'll have to cross across the capillary into that extracellular fluid, and then into whichever cell they're delivering the oxygen to, whether it's a muscle cell or anything else. So the red blood cells never actually leave the blood vessel, but white blood cells do. So white blood cells will actually leave the circulation. They'll move through the capillary wall. They'll move into that loose connective tissue. They'll move into that extracellular space. And in fact, one of the things that they're able to do is actually change shape a little bit, and to sort of grow cellular extensions that act to help them swim through that extracellular fluid. And they're attracted to inflammatory chemicals, things like histamine, that are released by cells like your mast cells, which we talked about a little bit when we talked about the inflammatory response. But also the basophils there on the bottom release some chemical mediators that attract other white blood cells as well. And so basically they follow this this sort of breadcrumb trail of inflammatory chemicals that attracts them to the area so that they know where the inflammation is. And interestingly, in the endothelium of your blood vessels, where there is inflammation, the endothelial cells sort of sprout these extensions, kind of like sticking a hand out, that show the white blood cells that this is the area where there's inflammation. So then the white blood cells will attach there. They'll move from the circulation across the blood vessel and then into that extracellular tissue. So the first one on there, your neutrophils. So those are the ones we have that comprise the vast majority of our white blood cells. They're somewhere between 50 and 75% of our white blood cells, and they're pretty big. They're about twice the size of your red blood cells. And one of the things to know about them is they are our primary bacteria slayers. So they're the ones that are going to be involved in killing bacteria that get in, through a cut, or that get across the endothelium, or not endothelium, the epithelium of the small intestine, those kinds of things. So the neutrophils, and the way they do that, they're phagocytic. They're able to essentially absorb that bacteria. They envelop that bacteria, and then once they get the bacteria inside of them, they have lysosomes, the stomachs that they can use to digest it. But they also have granules that have the ability to essentially pierce the cell membrane of that bacteria. So they can put a bunch of holes in the membrane of that bacteria. And then neutrophils, interestingly, also produce bleach or hydrogen peroxide. And so then they're able to kill those bacteria. So neutrophils are really ruthless killers of bacteria, which is good. So other things about them, we don't really get into the immune system, but there's essentially two components to the immune system. There's the innate immune system and then the acquired immune system. So the innate immune system is like the neutrophils that they see bacteria and they attack them. And then the lymphocytes are part of the acquired immune system. So the acquired immune system is the part of our immune system that is responsive to things like vaccinations. So when you get exposed to something like the flu virus, then your immune system recognizes the particular nucleic acid sequence of that virus, and so the next time it sees it, you ramp up production of the white blood cells really quickly and you attack it really quickly. And so that is your acquired immune system. You've acquired it after being exposed to that particular pathogen. So the lymphocytes then are part of that acquired immune system, and they comprise the next highest concentration of white blood cells. So the lymphocytes make up somewhere in the neighborhood of 25% of your white blood cells. And they exist, there are some of them in the blood, but they exist primarily in the lymphatic vessels. So again, lymphatic system returns blood back to the central, or not blood, sorry, lymphatic system returns fluid back to the central circulation. And then the next one down there, monocytes, those are somewhere between 3 and 8% of our white blood cells, and those are going to, once they get in the tissue, they're going to differentiate into macrophages. And we talked about macrophages with inflammation, that those are the big eaters. Those are the ones that not only attack bacteria and viruses, but they also have the capacity to chew up damaged tissue. So if you get, if you get a cut, you're going to have some collagen strands that have been torn as part of that. And so the macrophages are the ones that help sort of chew up those ends of the collagen sort of complete that breakdown process before we make some new tissue there. Then you got the eosinophils. I think I pronounced it correctly. I'm close anyway. And so those, in terms of concentration, they're next one down on the list. So they make up about 2 to 4% of our leukocytes. So they also have digestive enzymes, but they lack the ability to kill bacteria. And instead, those are the ones that play a role in if you get worms. They're good at killing those. So either worms that burrow through the skin or through the digestive tract. So things like tapeworms, the eosinophils, eosinophils. Those are the ones that attack those. They use their lysosomes. They use their granules to they basically merge with the worm and then attach their little digestive enzymes to it and break down the surface of the worm to kill it. And then the last one on there, the basophils. The basophils essentially signal the arrest of them, so they release the chemical mediators that that attract the neutrophils and the lymphocytes to the area. So the basophils make up less than 1% of your white blood cells, and they act very similar to the mast cells in terms of signaling, hey, this is where damage is occurring, and so they stimulate other white blood cells to move to the area. All right, so that's blood. So then we'll move on to the heart next time. So we'll see you then.